you guys aren't and you can keep me awake. So we're right here in the middle of the central metabolic pathways, and we've talked about them a bit, and we're kind of cycling back through and looking at them a little bit more in detail. And remember, I told you the pentose phosphate pathway last time used to be called the pentose phosphate shunt. Do you remember why scientists back in the 60s, 70s, 80s, 90s called it the pentose phosphate shunt instead of the pentose phosphate pathway? Why did they consider it a shunt? You know what a shunt does? A shunt redirects something, and so this used to be called the pentose phosphate shunt. Do you remember why? What feeds into the PPP? Did any of you go look at that appendix at the back? I usually encourage you to look at appendix. I think it's appendix 2. Sometime you can flip back there now if you want, but sometime outside of class, go look at it. It's really complicated. It is the flow of the pentose phosphate pathway. And there's like 3 cycles in the middle and a bunch of side cycles, and it's a really complex pathway. And back when I took microbiology in 1979-80, we had to memorize that. And so when students complain that there's too much to know, I direct them to that pathway and say, look, we used to have to know this much detail when we took this freshman, sophomore level course, and all you need to know is the name and what the products are that come out of it now. And it's an amazingly complex pathway. The reason it was called a shunt is because the first product out of glycolysis gets shunted out of glycolysis into the pentose phosphate pathway. What's the first product of glycolysis? Let me just see what you remember from last time.
Glucose 6.
Glucose 6 phosphate. There's a phosphate on the number 6 carbon, so we call it glucose 6 phosphate. And it gets pulled out of glycolysis to go over into the pentose phosphate pathway. And it can be converted into ribose 5 phosphate. Ribose is a 5-carbon molecule. That means the glucose 6 phosphate lost 1 carbon and was converted into ribose 5 phosphate. The other thing that's important, or one of the other things that's important that comes out of this pathway, is erythrose 4 phosphate. And erythrose 4 phosphate can be used in one type of biosynthesis. Do you remember what kind of biosynthesis that is? Yeah, you guys are about as awake as I feel this morning. Protein biosynthesis. It's used to synthesize amino acids. So it's the substrate to make a couple of amino acids, including histidine. And let's go backward. The ribose 5 phosphate is also used in biosynthesis. What kind of biosynthesis do you think it would be used in? Nucleic acid biosynthesis, which includes what types of molecules? RNA and DNA, right? So all the RNA in your body, all the DNA in your body starts with the carbohydrate part of it as ribose 5 phosphate. That oxygen's removed, and then it becomes deoxyribose for the DNA. The other important product that's produced out of the pentose phosphate pathway is this NADPH. And NADPH, we've talked about, is used in what kind of reactions? Yeah, there's going to be some redox happening because it's going to donate electrons. It is an electron carrier, right? It's needed by many enzymes as a cofactor. Oh, I said that wrong. It's needed by many enzymes as a coenzyme. What's the difference between cofactors and coenzymes? Pretty sure we talked about that last time. Cofactors are inorganic molecules that are needed by enzymes to do something, either donate or accept an electron. And coenzymes are organic molecules that are needed by enzymes to either donate or accept electrons. So if NADPH is a coenzyme, what do you think it does when it works alongside of that enzyme or those enzymes that it's going to help facilitate their function? It's going to donate electrons. So if it's donating electrons in an enzymatic reaction, is that a reduction reaction or an oxidation reaction? If you're talking about the substrate, not the NADPH, which is it? The substrate's receiving electrons. So what do we call that? Reduction reaction. So NADPH and all of those electron carriers that are housing those electrons, we say they have reducing power, right? That means they have the ability to reduce something else. And in this case, NADPH has reducing power, and it typically functions in biosynthetic processes. It's going to drive enzymes. And you can starve enzymes. If they need NADPH and you just give them a little bit, they'll work for a little while. And if you just give them a little bit, they'll probably work slowly because they need the NADPH for the energy to drive the function of that enzyme. And if you don't give them very much, as soon as they run out or can't get access to it, the enzymes will slow down. So you can change, we've looked at some enzyme curves and some function of the curves. You can change the function of enzymes, but not giving them enough of that coenzyme that they need to donate electrons. So what do you think about this question? Can NADP function as a coenzyme with another enzyme to drive or facilitate a reaction? Can NADP? Yes, why do you think so? What kind of reaction would that be? Yeah, that would be an oxidative reaction. In other words, the substrate would be giving off the electrons. NADP would be receiving those electrons, and it would be converted to NADPH in the process of that particular reaction. So you can see it. And if they're serving as a coenzyme and you're looking down in with an electron microscope, where would you expect to find them in that complex that's doing those reactions? In the active site of the enzyme. Good. You'd expect to see them right down inside of the active site. Beautiful. Just want to review a few things to make sure you got some of the principles that we've already talked about from previous parts of this chapter, and to just wake you up just a little bit. All right, so we're back in the central metabolic pathway, and we're going from pyruvate. And where did that pyruvate come from? Glycolysis, right? The end product of glycolysis is 2 pyruvates, and pyruvates are 3 carbon molecules. You take a 6 carbon molecule and you break it into 2 3 carbon molecules, and then that's how that happens. That's what glycolysis does. It releases generates a little bit of ATP, generates a little bit of NADH, and you get 2 pyruvates coming out of it. We need a way to feed pyruvate into this cycle, and you guys know this cycle by 3 different names: the TCA cycle, the Krebs cycle, and the citric acid cycle. And so we need a way to feed it in, and that's through this one-step reaction right here that's called the intermediate step. And the intermediate step basically kicks out a CO2. Remember, that's what respiration in general does, is kicks out a CO2, and you can see that CO2 molecule coming out right here. And that CO2 is kicked out. We get this coenzyme A coming in here. It's attached to 2 of the carbons from the pyruvate. The 3rd carbon leaves as carbon dioxide. Everybody with me? I see some of you exhaling, so keep kicking out that carbon dioxide so you can stay awake. And so now we've got this product of the reaction. The pyruvate's converted to an acetyl group that's attached to this coenzyme A. Okay? So coenzyme A, you need to do this. Make a note to self so you can recognize it on a test down the road. Coenzyme A is actually a very large molecule. They write CoA there, and it makes it look like it's almost nothing because you've got 2 carbons attached to it. The 2 carbons actually look bigger, but coenzyme A is a large molecule. I want you to go to Wikipedia because it's the easiest place for you to find the information. I know this page is accurate. Go to Wikipedia and look at coenzyme A. Find the structure, count the carbon number, and bring that back to class tomorrow. And tell me how many carbons. Go ahead and write it on just a small slip of paper so I can go ahead and collect that as a class participation grade tomorrow. So go to Wikipedia, look at the structure of coenzyme A, leave the two carbons from the acetyl group off, just coenzyme A, and tell me the structure and how many carbons are in that molecule when we come to class tomorrow. It's a big molecule. I want you to look at it so you can visualize this. It's functioning as, what's its name? Yeah, what's the CoA stand for? Coenzyme A. It's a coenzyme. And what did I just ask you? What do coenzymes do? They have to get down into the active site of the enzyme and facilitate that reaction. So it's a coenzyme. We call it coenzyme A. It's got to get into the active site to facilitate this particular reaction. If there's no coenzyme A, this reaction won't go. Yes, sir?
You wanted the structure and what else?
How many carbons are in that coenzyme A? Okay. So anyway, coenzyme A gets linked to the 2 carbons from the pyruvate, and now we're right down here. And so the things that come out of the transition step are a CO2, and then we get this reduction reaction that produces NADH. So we get some more reducing power out of this transition step. We get some energy that can drive another reaction. Or we get some energy, in this case, it's NADH. Where can NADH go besides being used to drive some biosynthetic enzymes? Where can it be used? It can be, it's got electrons, it's a carrier, or yeah, it's an electron carrier. What do you think that stands for? Whoops. Electron transport chain, or some books call it the electron transport system. So that's where those enzymes can go, but NADH can also work as a coenzyme for some biosynthetic enzymes. So if we look at this step, the transition step, did you notice what happened in this step? What could we call NAD+? It's an acceptor, right? It's an acceptor of electrons in that particular reaction. But it's a organic molecule, and it's another coenzyme for this particular enzyme. This enzyme will also die if you don't feed it NAD+. It has to have NAD+ to be able to drive this particular reaction. So it's another coenzyme for that particular reaction. Okay, so we get 2 carbons attached to an acetyl group. Those 2 carbons come and are bound to 4 carbons in this molecule that's called oxaloacetate. And then those 4 carbons, it's some pretty simple math, get connected to 2 carbons. That makes 6 carbons. And that's the first product that comes out of the TCA cycle. And that's why sometimes it's called the citric acid cycle. The first product is citrate, and another name for citrate is citric acid. So citrate then gets converted to isocitrate in a simple rearrangement step, and then look what happens next in the next step. The citrate is converted to alpha-ketoglutarate. How many carbons does alpha-ketoglutarate have? 5 carbons. So what happens to that extra carbon? It gets kicked out, part of the respiration process. Again, it gets kicked out as CO2 in that reaction. And look what happened also in that reaction. You got some NAD+ that's being changed into NADH. Is that an oxidation or a reduction reaction? Reduction, right? It was a positive charge. It picked up 2 electrons. When you pick up electrons, something's getting reduced, right? And so the NAD+ got reduced to NADH. It can go on over to the electron transport chain as well and drive the electron transport chain. Okay? And we got alpha-ketoglutarate coming out of that particular reaction. We're right here. Look what happens in the next step. Another CO2 kicks off. Alpha-ketoglutarate has how many carbons? 5 carbons. You lose 1, now you got 4 carbons. We got down to succinyl-CoA, and a couple things happen. These reactions are a little bit complicated. The enzymes need a lot of different cofactors. It needs NAD+, you can see that there, and it also needs coenzyme A there, and so both of those things have to feed into this reaction to drive that process. We just saw another coenzyme A coming in. It could be this one that came out here and feeds in down to here, or it could be another one that's floating around in the cell. That coenzyme A gets attached. Now we've got succinyl attached to coenzyme A. And again, coenzyme A is the carrier. The enzyme that's involved in this step needs to see the succinyl attached to that coenzyme. And then the book will tell you through a series of complex reactions, and if you want to know about it, you can go back and look at another appendix. It is a pretty complex process. You end up getting the ATP generated there. There's temporarily an inorganic phosphate that's added to the succinate. It's a high-energy bond. And that high-energy bond has enough energy so that the phosphate can then be transferred to ADP to make ATP. And when that happens, what kind of phosphorylation are we seeing occur right down here at the bottom of this process? We've got substrate level phosphorylation. How do you know it's substrate level phosphorylation? phosphate is put onto another molecule, a carrier, on a high energy phosphate bond, and it's transferred from that substrate that's carrying that phosphate to ADP, and that's always substrate level phosphorylation. Okay, so we get ATP being produced, 1 ATP per cycle. So I could ask you a test question down the road. If glucose feeds into glycolysis and it goes all the way through the TCA cycle and no precursor metabolites are pulled out to make other anabolic building blocks, how much ATP can you get from the TCA cycle if that glucose feeds all the way through? And if you only consider the ATP and not the reducing power, what would the answer be? 2, right? 2 from the TCA cycle. And how many from glycolysis, just to step back and make sure you tie it together? 2, right? You make 4 in glycolysis, but you have to feed 2 in to kickstart it. And so you end up making 2 in glycolysis, 2 in the TCA. But now we've got a whole bunch of reducing power out of these systems. So let's look at that. Remember, we got 1 NADH. We got 2 from glycolysis. We got some NADH here. Let me say this. We got some NADH here. We got some NADH here. And we got some NADH here. And we also got FADH2. And all of those, if the organism has a functional electron transport chain, can feed in there and drive that electron transport chain. But remember, now we're talking about some bugs that are very different from our cells, and many of them don't have an electron transport chain. Many of them can't use that NADH and FADH2 to drive that electron transport chain, and if they can't, they have to do something else, because each one of those reactions that I just circled needs the oxidized form to be able to go. And if the cell runs out of the oxidized form because it's constantly reducing it, all of those enzymes that are marked by those circles are going to stop, and the cell won't be able to do those processes anymore. Question?
Don't we run the Krebs cycle twice?
Exactly right. So we get 2 ATP down here at the bottom, 2 for each glucose that goes through. If the question asks, per cycle of the TCA cycle, how many ATP is produced, and probably I'd be specific and say by substrate level phosphorylation, then the answer for that per turn of the TCA cycle is it's 1 ATP. Okay? If I didn't specify via substrate level phosphorylation, then you'd probably want to do some quick math and start to say, okay, there's 1 NADH that's produced, there's 2, there's 3 NADHs that are produced over there, and for each NADH, if it can feed into an electron transport chain, there are 3 possible ATP that can be produced out of each one of those, and then the FADH2 can make 2 more, so you'd want to do some quick math and see if that combination of possible number of ATP is represented in the question. Because if the organism, like your mitochondria, has the electron transport chain, you can actually get more ATP, but it's not coming directly from this TCA cycle. It's coming indirectly by feeding all of those electron carriers into the electron transport chain. Okay, so do you remember why these guys are in boxes? Do you remember why those are in boxes? Nope. They're precursor metabolites. Do you remember what a precursor metabolite is?
Intermediate.
Yeah, it's an intermediate that comes out of these reactions, but precursor metabolite. Metabolism does 2 things, right? What are the 2 aspects of metabolism? Anabolism and catabolism, and we're in the middle of catabolism here. So if these are precursor metabolites, what do you think they might do? They might just feed out of here into a different type of metabolism, which we call what? Anabolism, right? All of those structures that are in the boxes are precursor metabolites. It's true for glycolysis too. All of those that are in the boxes are precursor metabolites. That means they can be used as building blocks to make those macromolecules that we talked about in chapter 2. And if they are used, for example, if the pyruvate, I'm pretty sure I mentioned it last time, if the pyruvate is pulled out of here and taken to make amino acids, and it can make 5, 6 different amino acids depending on the cell, if it's pulled out of there, that pyruvate, the carbon, never feeds through this cycle. And that means none of those products are ever made from that cycle. That means none of the ATP comes out. That means none of the reducing power comes out if the pyruvate is pulled out to make amino acids out of that, the end of glycolysis, before it actually gets into the transition step. The same thing will be true for alpha-ketoglutarate. If the alpha-ketoglutarate gets pulled out, and it goes into amino acid biosynthesis as well, if it's pulled out, then again, this ATP doesn't happen. This FADH2 doesn't get produced. This NADH doesn't get produced, and those carbons no longer go around and can start another cycle because those carbons got pulled out of the pathway. Does that make sense? Okay, cool. So you lose all of the energy that those molecules are carrying if they're pulled out as precursor metabolites to be used as building blocks for other macromolecules. So the acetyl-CoA gets pulled out. Actually, it's the acetyl group that gets pulled out up here. And I'm just curious if you remember from anatomy and physiology, what's made from those 2 carbons? What was it? Some of them might be made, but there's a general class of macromolecules that are made from those 2 carbons. Lipids, beautiful. Those 2 carbons are pulled out for lipid biosynthesis to synthesize which part of the lipids, since lipids are a complex molecule. There's 2 parts to lipids, right? What are the 2 parts to, if we just talk about a simple lipid? Fatty acids is one, and glycerol. So which one of those 2 components do you think the acetyl group goes into synthesis of? The fatty acid side chains, if you look at them, they're almost all multiples of 2 in length. 10, 12, 14, 16, 18, 22. And the reason for that is because these 2 carbons feed into their biosynthesis. There's a few that get enzymatically clipped, so you get an odd number, but most all of them are going to be multiples of 2 in length. And it's because of the synthetic process, the way that they're made. Those 2 carbons right there get pulled off. And if that happens, this cycle of the TCA step never goes. It's stopped at that point. Everybody good with that? Okay. All right, good deal. So the transition step makes a couple of products. We've talked about this before in our cells, and in eukaryotic cells, the TCA cycle and the transition step happens inside of the mitochondria. And that's part of the reason you get less energy from glucose in an ATP cell, because you've got to transport the product from glycolysis, the pyruvate, into the mitochondria and that takes energy for the cell. And so because prokaryotes are less compartmentalized, we can get more energy out of one glucose molecule in a prokaryotic system. There's no need to transport the pyruvate across that mitochondrial membrane. It's essentially already inside of a cell that functions a lot like the mitochondria, except for the cytoplasm now has all the enzymes to do glycolysis and all the enzymes to do the TCA step. So the TCA cycle completes the oxidation process of glucose. What in the world does that mean? The TCA cycle completes the oxidation step of glucose. So the first thing you have to ask yourself is, what does it mean for glucose to be oxidized? And how does it happen? What is the main product when glucose is oxidized? What do you think? What's glucose, the majority of glucose made out of? Remember, I think I taught you this Chinese term last time, right? What does this mean? It means 6. 5 plus 1 is 6. Okay? 6 carbons. Glucose has 6 carbons on it. And what do you see coming out of all of these steps? I don't want to go all the way back to glycolysis, but glucose, the oxidation of glucose begins in glycolysis, right? And if you look at that process, the glucose is being broken down, and now we're seeing the oxidation of those carbons that came from glucose as they feed into this cycle. We're getting them oxidized, and when they're oxidized, they're picking up oxygen, and they're being released as CO2. So that's what this term complete oxidation of glucose means. There's oxidation steps that are going on all the way along. Look back and you'll see those electron carriers, the NAD that's feeding into glucose or glycolysis, they're getting reduced. That means glucose is being oxidized, right? By definition. So we get the oxidation, and what that means is that we've got CO2 coming out of this process in those three places. That was four places. Where did I miss the fourth one? 1, 2, 3. I'm wondering if my slide's not wrong. I need to check on that. So,
Well, isn't it?
yeah, anyway, it oxidizes glucose. You also get from the TCA cycle 2 ATP. Those happen down here. And what's the process that produces those ATP? Starts with the S. Substrate level phosphorylation. Beautiful. And we get 6 NADH, and that number again is per glucose. 6 NADH, 2 FADH2. So you could take both of those numbers and talk about them per cycle or per turn of the TCA cycle, and it would be half of that, right? Because glycolysis feeds in 2 pyruvates up here, and so this cycle is going to go twice instead of just once if we're talking about glucose feeding in. All right, and then we get precursor metabolites, and I'm pretty sure you want to know those. Some textbooks separate these two steps, the intermediate step and the TCA cycle, and sometimes here you see that when they're talking about the products that are produced, they're actually talking about them differently because they, oh, I get it. Now I know why there's 4. Did you guys figure it out yet? I was scratching my head up here, but it just took me a second to figure out why there's 4. There's 2 here, 1 here and 1 here, and if it goes around twice, it makes this one 2x, and it makes this one 2x. Everybody with it now? Sorry, my slide's not wrong. And it just took my brain a second to wake up on that. So the precursor metabolites, again, are these guys that are in the boxes, and all but one of those feed into protein biosynthesis. Which one of those 4 precursor metabolites does not feed into protein biosynthesis? We just talked about it. It's this guy. And what kind of biosynthesis does it feed into? Lipids. Okay. So all 3 of the others feed into protein biosynthesis. And it feeds into lipid biosynthesis. Okay, so now we need to switch gears a little bit away from the central metabolic pathways and talk about the process of respiration. Respiration basically is using all the reducing power, all that NADH and FADH2 that's produced in all of those central metabolic pathways, and we use it to synthesize ATP. Why are those molecules called reducing power? They're the products of redox reactions, and are they the reductant, or are they being reduced, or are they being oxidized in those initial redox reactions that they're products from? They're being reduced, and these are the reduced form. If these are the reduced form, what do you intuitively know about these molecules, NADH and FADH2? If they're the reduced form, what can you tell me about them? They're carrying 2 high-energy electrons, right? They have 2 high-energy electrons that are buried down inside of those letters. You can't see them, but if they're the reduced form, when reduction happens, electrons are transferred to that molecule. So NADH and FADH2 have 2 high-energy electrons, and they're called electron carriers. We talked about that last time. They're being carried over to the electron transport chain. In this case, as we're talking about respiration, and they're going to be used there to synthesize ATP. They're not used directly to synthesize ATP. It's indirect, but the electrons they carry are driving the systems that are able to run and produce ATP. The enzyme that actually produces the ATP, you should probably know this from anatomy and physiology, it's called ATP synthase. That enzyme named synthase makes sense. It synthesizes something, tells you it's an enzyme that synthesizes something, and the first part of the name actually tells you what it synthesizes. It synthesizes ATP. So ATP synthase is a very logical name for this particular enzyme, and ATP synthase can use the energy that's produced from these guys charging the electron transport chain, because you know that those guys, those electron carriers, charge the proton pumps that pump protons across membranes. You know that from anatomy and physiology, right? And those electrons are charging the pumps. They pump protons out of the cytoplasm across that membrane and make a proton barrier. And that proton barrier we call a proton motive force. And the proton motive force is what actually drives this enzyme, ATP synthase. So ATP synthase basically uses the electric current, if you will, except for it's not electrons flowing, it's protons that are flowing across a membrane, and those small positively charged particles that are flowing across the membrane then function to provide the energy that ATP synthase needs to phosphorylate ADP and make ATP. Everybody with me on that? Okay. You already knew all of that from your anatomy physiology classes, and if you had figured it out earlier, you probably could have got a Nobel Prize for figuring out how all that stuff worked. Come on, projector, work with me. So this is what the electron transport chain does in simple terms. It takes high energy electrons and it passes them from step to step to step to step to step until you get down to the bottom. And in our case, in the mitochondria, those low energy electrons, because their energy's been donated along the way as they're driving this electron transport chain in those proton pumps, but they're still electrons. They got to go somewhere. And in our case, we need oxygen because our mitochondria need oxygen as the final electron acceptor for this electron transport chain. I'm sure you remember that from anatomy and physiology. That oxygen picks up the electron that comes along and it finds two protons, two hydrogen in the environment, and it turns out it makes water. And water is produced in this reaction. And I think you already knew that. So this electron transport chain in the mitochondria is in the inner membrane of the mitochondria. In prokaryotes, these same or very similar enzymes sit out in the cytoplasmic membrane. And they pump the protons across the cytoplasmic membrane from the cytoplasm to the periplasm if it's a gram-negative, and from the cytoplasm to just outside of the cytoplasmic membrane if it's a gram-positive. The whole process gradually releases the energy that those highly charged electrons bring into the system. And as it gradually releases, it ejects protons from the cytoplasm. It creates an electrochemical gradient, protons in high concentration on one side, low concentration on the other. Long ago we talked about the pH of the periplasm, and we said it was acidic. And the reason for that is these protons that are being pumped out there into the periplasm. It makes that periplasm an acidic environment. And that gradient then is used to synthesize ATP by that enzyme ATP synthase. In prokaryotes, it does a couple of other things as well. Powers some transporters. In anatomy and physiology, you talked about some molecules being brought from outside of the cell, inside of the cell by active transport. Do you remember that? And when in active transport, the way you talked about it in anatomy and physiology, glucose is one of those molecules that's brought in via active transport into the cell. Some of it can diffuse in, but there are some specific carriers that sometimes need energy. Do you remember what provides the energy for those molecules to be carried from outside of the cell to the inside of the cell when we're thinking about eukaryotic cells? ATP is the energy source that drives that. But there are some of those transporters in prokaryotes that can actually use this proton motive force as the energy force to pull that in. And if you look back to one of the previous chapters, we already actually talked about that, so you should remember that. The other important thing that this proton motive force is used for, if the organism has a flagellum or flagella, then the proton gradient is used to drive those flagella for motility. One of the cool things you're going to do in lab this week is you're going to get a special kind of agar. That agar has all the nutrients that all the rest of them that you'll work with have. Plenty of nutrients for everything that we work with to grow. The only difference between it and the other type of agar that you'll use in a tube, or the main difference, is that the agar concentration is about 50% or 30% of the normal concentration. And you'll take that other inoculating tool that's in your drawer. You might have noticed that it doesn't have a loop on the end. We call it an inoculating needle. And you'll take that inoculating needle and you'll sterilize it, and then you'll go into the tube and get some organism, and then you'll stab right down the center of the agar that's in the tube, and you'll look to see after 48 hours if the organism moved or if it didn't move. What you're really looking for is to see if it has the ability to produce a flagellum. When you put it in that agar and stab it in there, just straight in and back out again, the organism is placed in one place, and it'll start munching the nutrients that are there in that agar. But pretty soon it's going to get hungry because all of its friends are growing, and it's making kids and babies and getting millions of cells down in there. If you took that to a dissecting microscope or your microscope, you could look down and you'd see little colonies right along the stab line initially. But as they start to get hungry, they say, uh-oh, we're running out of nutrients. We need to get someplace else if they can. And if they can, they'll synthesize their flagellum, and they'll start to do that process called chemotaxis. Do you remember what chemotaxis is? Moving across a chemical gradient. They just used up all the available nutrients, so there's low nutrients, and they start moving to areas that have higher nutrients. In other words, they're moving away from the stab line. And you're looking to see if they can move or not. Some of them that you look at will be able to move. In other words, they have a flagellum or flagella, and others won't be able to move. If they can't move, you put them right along the stab line, and they're stuck there on that stab line, and you'll actually see little beads along the stab line where they form colonies down inside of the agar because they can't produce a flagellum. They can't swim away from that area. If they are motile, if they can produce a flagellum, they'll actually swim away from there pretty quickly, and all you'll see is kind of a haze moving away from the stab line as the bacteria move out to find the fresh nutrients. And they're doing that, and in doing that, they're using energy from the proton motive force that we're talking about here to be able to swim away. Okay? So they'll use that proton gradient that they're generating as they're doing this kind of metabolism to be able to swim away. So the electron transport chain has a number of components, and basically we've got carriers that are feeding in, and they feed into large protein complexes, and most of these guys serve as proton pumps. What does that name tell you they probably do? They move electrons. They move electrons, and I just told you from where. Where do they move the electrons? Oh, sorry, they don't move electrons. They move something else. I lied to you. You tricked me.
Protons.
They move protons, right? And where do they move the protons from? Inside of the cytoplasm to the outside of the cell, right? So they're just pumps. Your stomach has beautiful proton pumps. You eat a nice meal, put lots of proteins, put lots of carbohydrates in there. Those all raise the pH. Your stomach says, hey, you need to do something about this. Proton pumps kick on. They start pumping protons into the lumen of your stomach to reduce to lower the pH and reduce the available protons in that environment. The same type of thing is happening here. We're moving protons. And so there are three general groups. One are the quinones, and these guys are lipid-soluble molecules. They can move freely around. They can transfer electrons between complexes. The second one is the cytochromes, and these guys typically contain heme or other heme-like molecules. That means that they're proteins that have iron down at the center. There's a number of types of those guys. And then there's the flavoproteins. And these guys have flavin, one of the coenzymes that is necessary. And FAD or FADH2 will be a coenzyme for these guys. Okay? And so we're going to look at the electron transport chain because I'm 99% sure you remember almost all of it from anatomy and physiology, but I bet you there's one of you in this class that's forgotten some of the information about the electron transport chain from anatomy and physiology. And so we need to look at it in general terms, and then we'll go look at it in specific terms. So the general mechanism basically ejects those protons. We've got some carriers that accept hydrogen and electrons in pairs. Can you name those guys? Which ones feed into the electron transport chain? NADH is one, and FADH2 is the other one. So they carry protons and electrons in pair. Some of the others only carry electrons, and it just depends on what system you're looking at. But the systems that we're most familiar with, including the mitochondria, they're going to carry 2 electrons and usually at least 1 proton along with that. And usually there's one that's tagging along the second electron. So they pick up protons from inside of the cell. Protons are hydrogen, and then they pass the electrons to the electron carrier, and then the protons are released on the outside of the cell. That's what a proton pump does in specific terms. Pumps protons outside of the cell. The protons are carried there by the electron carriers, and then they're moved outside of the cell. The electrons that those electron carriers are bringing drive those proton pumps. And that effect is we get a proton gradient across the membrane, and the energy that's used drives that process. So we can look at the mitochondria, but I know you already have. What I want you to do is look at the figure for the mitochondria and get familiar with that. Actually, you're already familiar because you looked at that in anatomy and physiology. And so you know this process from A&P pretty well. If you don't, you want to go and review the process that happens down inside of your mitochondria. But it's pretty analogous to what happens in E. coli, and that's what we're going to look at. If we look at E. coli when it's doing aerobic respiration, E. coli does respiration almost exactly like our cells do in the aerobic environment. And basically, it can use 2 different NADH dehydrogenases as proton pumps. And these guys, if you compare the mitochondria, are equivalent to that complex 1 that you learned about in the mitochondria that's also a proton pump. And then E. coli also uses an enzyme called succinate dehydrogenase, and it's equivalent to complex 2 that you learned about in anatomy and physiology in the mitochondria. Basically, they're analogous proton pumps. They do the same thing. They sit in the same positions of the 2 electron transport chains, and E. coli doesn't have that complex 3 or cytochrome C. Some other organisms do have it, and that's another test you'll actually do next week in the microlab to see if they have an enzyme that can deal with cytochrome C called cytochrome C oxidase. All of this is tying together with things that you're going to be doing in the lab in the next couple of weeks. Okay? But the whole purpose of each one of those is to accept electrons, pump protons, and move the electron, once that bit of its energy is used, on down the chain to somebody else that now can use that energy that's lower from the process. And so here's the E. coli electron transport chain. Remember, is E. coli gram-positive or gram-negative? Gram-negative, right? And so if it's gram-negative, it's got that space that's in between the 2 membranes that's called the periplasm, or the periplasmic space. And so basically, if they drew this picture more accurately, they would put another lipid bilayer out here, right? And we've got these proton pumps that are along the way here that are, oops, that's not one. Proton pumps along the way that are pumping protons outside of the cell into the periplasm. And the end process is we get a gradient of protons out here in the periplasm, And those protons can be used to drive this enzyme called ATP synthase to ultimately take ADP and phosphorylate it into ATP. Everybody good with that? That whole picture you've seen before, at least as a significant part of one other class. This whole process is sometimes, the ATP that's produced from this process is called, it's produced by oxidative phosphorylation. I used to do a survey to see if students could tell me why it's called oxidative phosphorylation. What do you guys think? Look at the end product. The electron goes from this last pump, it's received by oxygen, and water's being produced. So you get water out of it, you get ATP out of it. Why do we call this process oxidative phosphorylation?
It's because the accepting the end product is H2O, there's the oxygen.
What do you guys think? Yes or no? Any other plausible answers?
Because protons are being produced and it's going to be needed for the production of ATP.
Protons are produced. The energy from the proton gradient is needed to drive that enzyme. And so, because protons are being used, we're going to call it oxidative phosphorylation?
Oh, it's a reduction, correct?
Yeah. So should we call it reductive phosphorylation?
No, but the proton acceptor in this case would be the ADP. So therefore, it is an oxidative reaction since it's negatively charged.
I tricked you a little bit. Because students usually don't know and don't ask why it's called oxidative phosphorylation. And so I made you come look over here. And that's what students usually guess, because oxygen's involved in that final step as the final electron acceptor. But I misled you away from the answer. The answer is actually over here. What's happening to the NADH at the beginning of that process? NADH is being oxidized to NAD. And I can't write it out here with this pen because it's too small, but FADH2 is feeding in right here at succinate dehydrogenase, and then it comes out as FAD+. In that case, FADH2 is donating electrons. What's happening to the FADH2? It's being oxidized. So now if I asked you, why do we call this oxidative phosphorylation, what would you tell me? The very beginning of the process requires oxidation reactions. There's one important one that's going on right here, and another one I just drew in here, but if you read the text below this figure in the book, they'll tell you about how FADH2 feeds into succinate dehydrogenase and is oxidized by succinate dehydrogenase at that step. Okay? And both of those are oxidation reactions. So the final product that comes out of this process, this ATP right here, we call it being produced by oxidative phosphorylation, because what kicks it all off is an oxidation reaction. The energy that drives the process is a series of oxidation reactions. And if those oxidation reactions don't happen, we don't get this ATP being produced over here. All right, cool. So I taught you something, you know a little bit about that. So we've got two proton pumps that are involved. The first one is called NADH dehydrogenase. And if FADH2 is feeding in, if NADH is feeding in, that's where it'll go. It'll go into this proton, which takes the electrons, and in the process pumps the protons out into the periplasm. Those electrons drive that enzyme, but they still have a little bit of energy. So they get transferred over to this small molecule called ubiquinone, and ubiquinone accepts those electrons. It absorbs a little bit of their energy, but then it passes the electrons over to this next proton pump that's called ubiquinol oxidase. And ubiquinol oxidase accepts those electrons, and it pumps some more protons from the cytoplasm out into the periplasm. Those electrons are still there, they're just now lower energy, and those electrons then, if oxygen is around, and for your mitochondria, oxygen has to be around, and the oxygen will accept that low energy electron, and then protons will again be removed from the cytoplasm as water's produced. So I don't know if you ever thought about why you die, when you die, but this process is pretty essential to why you live. And if you suffocate, meaning that you don't have enough oxygen, then it's this process right here that's the reason that you die. In most all cases, a person dies when they stop breathing. And when you stop breathing, you're not replenishing the oxygen in your body. And when you're not replenishing the oxygen in the body, this oxygen right here is no longer available, which means your cells can't make any more ATP. And when they can't make any more ATP, pretty soon they die. There's some other contributing factors. The CO2 level builds up, and that's also toxic to the cells. But one of the main reasons that you die and that your cells die is because this process right here can't happen anymore. If this very process can't happen, this can't happen. And this can't happen because everything backs up, and now you've got all kinds of electrons. They can't go anywhere, and now you got a system that's really messed up because the electrons can't flow through that system. And in the case of your cells, the mitochondria stops working, and then that cell dies. If you're a prokaryote, it's the same thing, except for the whole cell dies because this is happening out at the cytoplasmic membrane. But some prokaryotes figured that out, I guess, or God figured out when He made them, and not all of them use oxygen as the terminal electron acceptor. Some of them have systems that can use other inorganic chemicals, and I mentioned that last time too, to be able to be that terminal electron acceptor. Okay? And if that's the case, usually, remember I told you this number, if it's a system that's got oxygen, I told you we can get 38 ATP out of it, right? But if there's no oxygen around, we can't get this going. And typically then we only get 36 ATP out of that system when we're talking about prokaryotes. That's the maximum we can get. What do you think's the difference between that 36 and that 38 if there was oxygen around? What's the difference? Why can you get more if there's oxygen around? In other words, why can you get more in the aerobic situation from the anaerobic situation?
So those two electrons that oxygen is accepting?
That's part of it. What's going on right here? How does this whole system work? How does this system work?
You get less energy.
You do get less energy, but the question is why? How does this whole system work? How does it drive ATP synthase? How does it drive these transporters? How does it drive the flagellum? How does it work? You have to get lots and lots of protons out here. Lots and lots and lots. And very little protons in here. I can't draw a small h, but very few of them down inside of here, and that makes a battery.
It's a concentration gradient.
It's a concentration gradient. It only works if there is a concentration gradient. And in chapter 5, we talked about some of those chemicals that infect or affect microbial growth, and some of them function as detergents, and they disrupt this outer membrane so that it's leaky now, and those protons then will flow out of the periplasm, and the proton motive force is disrupted, and they can't work anymore. Okay? So back to that original question, you know ATP synthase. ATP synthase is involved in harvesting the energy to make that proton motive force. And that's used to synthesize ATP. We need that gradient to do that. The gradient has energy, but the energy in the gradient is released as the protons flow back through. ATP synthase actually allows the protons to flow down that gradient, and as they flow, an inorganic phosphate group is added to ADP to make ATP. And typically, about 3 protons need to flow through to produce 1 ATP. So we can calculate the yields, and most textbooks say 3. This one says about 2.5 ATP from the electrons that NADH release, and about 1.5, most textbooks say 2 for FADH2. Why the difference? They're both donating 2 electrons. Why does FADH2, if we go with whole numbers, why does FADH2 only give 2 ATP, and why does NADH actually generate 3 ATP? What do you got?
Probably the affinity.
I'm jumping backward if you haven't figured out. It goes back to this figure. What did you learn from the stuff that I wrote on this figure as we were going through? It's obvious that NADH feeds in right here, right? Where does FADH2 feed in? Do you remember? It feeds in right here at succinate dehydrogenase. Because it feeds in right here, when FADH2 feeds its electrons into the system, they miss this proton pump. And those protons, therefore, that pump would have pumped out, don't get pumped. Because the electrons feed in right here, they flow through the system this way, and that first electron pump, that NADH dehydrogenase, those protons don't get pumped outside of the cell because FADH2's electrons never hit that particular proton pump. Okay? So that's the reason for the difference between the amount of ATP that gets produced from each one of the molecules, those coenzymes that are electron carriers that can feed into this process. So if we look at aerobic respiration in prokaryotes, we can start doing a little bit of math. We get 2 ATP from glycolysis. Remember, that's net gain. If I ask you the question, how many ATP are actually produced in glycolysis, what would you tell me? 4. Because 4 are made. There's 2 places where 2 are made on the branches as they go through, but you need 2 to kick it started. And then there's 2 more that are made in the TCA cycle, and you better be able to tell me that those are all made by substrate level phosphorylation. And you better be able to tell me what substrate level phosphorylation is. What's a quick, easy definition for substrate level phosphorylation? Some substrate, in all of these cases, carrying a high energy phosphate, and it's able to donate that phosphate and transfer, an enzyme can transfer it from that substrate that's phosphorylated to ADP to make ATP. That's the simple definition of substrate level phosphorylation. There's a number of substrates that can do that. You've seen 3. There's 2 different ones in glycolysis that can do it, and there's 1 in the TCA cycle that can be used for substrate level phosphorylation. So out of substrate level phosphorylation from those 2 processes, we get a total net gain of 4 ATP. Then if we look at oxidative phosphorylation, we can actually get 6 ATP from the reducing power that came out of glycolysis. There's a great test question. What is the reducing power that comes out of glycolysis that can be used for the production of ATP by oxidative phosphorylation? What is that reducing power? NADH, made in 2 places in glycolysis. Go back and look at that. Okay? It's on the same place on the 2 different branches. And then we get 6 ATP that come out of the transition step. And again, this is reducing power that's produced in the transition step. Transition step. What is that ATP? Not acetyl-CoA. Not in the transition step. No FADH2 comes out of the transition step. It's NADH that comes out again. NADH is produced. Again, it's got to be the reduced form to have reducing power in these situations. And then out of the TCA cycle, we can get 22. And another trick question, what's the reducing power that comes out of the TCA cycle that feeds into oxidative phosphorylation? NADH and FADH2 come out, okay? You can go back and do the math, but out of all of that reducing power, we get 34 ATP that can be produced, assuming all of these numbers are assuming one big thing. It's an aerobic situation, so it's already given that oxygen is there. But if we're going to get 38 ATP out of glucose, what has to not happen?
Disruption of these enzymes.
All the enzymes have to be there, that's for sure.
Well, the pre-metabolites.
The precursor metabolites have to not get pulled out. So, in a little bit, you're going to see that glucose 6-phosphate gets pulled out a lot. It gets pulled out a lot to make a really big set of macromolecules that the cell needs a lot of. So if that happens, the cell already invested ATP to make glucose 6-phosphate. And every time it makes one glucose 6-phosphate, it's down an ATP. It pumped an ATP, it used it, it made ADP. But you're going to see in just a second that it needs a lot. And if that happens, then basically none of those 38 ATP are produced. What's the net gain if glucose 6-phosphate gets pulled out as a precursor metabolite to build macromolecules for the cell? What's the net gain then? -1. The cell has to invest in ATP to make glucose 6-phosphate, doesn't get any out. Okay, everybody good with that? All right. So this figure kind of shows you, again, those same numbers, but I would probably pay attention to the table because you'll notice that they show that you have to, at the beginning you get 0 ATP out, and then finally they're saying down here you get an ATP out, but that ATP actually comes from this very last step right here. So the arrows are not in the right place. Make sure you don't try to remember the location that products come out from this particular figure. You want to go back to the main figure for glycolysis and the main figure for the transition step and the main figure for the TCA cycle. But if you just need something visual to remember the total number of products that are produced, then you can get that from here. Substrate level gives you 2 ATP. The oxidative, or the reduction power, that can feed into oxidative phosphorylation gives you 6 ATP out of glycolysis total. The transition step can give you a couple via oxidative phosphorylation because NADH is made there. Out of the TCA cycle, we get in 2 rounds 6 NADH, 2 FADH2, and then we've got substrate level phosphorylation happening right there that gives us 1 more ATP. So this figure is basically saying the same thing the last table did, but in a figure form, because I know that many of you learn by reading and memorizing charts and tables. Others of you can remember better by looking at a picture and remember where those things come out. So there's some organisms that live a pretty sad life. They apparently make some people feel happy, but they live a pretty sad life because all they can get out of life is 2 ATP for every glucose. Take glucose, run it through glycolysis, convert it into pyruvate, make a little bit of NADH. Now they've stuck with NADH and they've got pyruvate, but they don't have an intermediate step, they don't have a transition step, and those guys we call obligate fermenters. That's all the ATP that they can get is those last 2 ATP out of glycolysis. They just made some pyruvate, so they have to do something else with the pyruvate. And what they do is in this figure, they take the pyruvate, and some of them take the pyruvate, and they'll do this type of fermentation, and they'll produce lactate or lactic acid. For those of you guys that are joggers, Do you know why your muscles sometimes cramp?
Lactic acid.
It's the buildup of lactic acid. So that tells you something about your muscles. When you start to exercise, if you're exercising vigorously, those muscles can't get enough oxygen. And so if that happens, then they pump things out this pathway. They start fermentation because, remember, the electron transport chain needs oxygen in your tissues. It can't run anymore because there's no oxygen. And so they start to build up lactic acid by getting rid of the pyruvate and making lactic acid. The real reason they do that is because they want to squeak a little bit more energy out of the glucose that's there. And the only way now that they can do that is by running glycolysis to pyruvate. The other thing that happens here for these guys, if we switch and talk about bacteria that don't have the TCA cycle, because pretty soon you're going to stop and breathe. Pretty soon you're going to start to do some oxygen exchange. Pretty soon there's going to get oxygen that's back down to your mitochondria. They're going to switch back over to oxidative or aerobic respiration, and then they'll kick on some of the other systems that they turned off temporarily because there wasn't any oxygen there. But if we're talking about bacteria and they do this process, they've got a real serious problem if they're obligate fermenters because they don't have the electron transport chain. And the only way they can get energy is through glycolysis, and they need that reduced form, NADH, to be oxidized back to NAD+. Why would those obligate fermenters need NAD+? All they can do is glycolysis. Why do they need NAD+? It's a coenzyme that's needed. I think it's step 6, if I remember right. It's needed to be there. That enzyme has to have it. It has to accept those electrons in that reaction, and it becomes reduced. And they need to take that reduced form that you're seeing right here and reoxidize it so they can run glycolysis just a little bit more to get a little bit more ATP out. Other organisms do fermentation differently. They may take pyruvate, and this is the one that leg cramps rarely make people happy. And I don't know if this type of fermentation does either, but some people seem to think that it makes them happier. The organism takes pyruvate, kicks a CO2 off. Now we went from a 3-carbon molecule to a 2-carbon molecule. Again, we've got the same problem that the organism's trying to deal with. All the NAD has been converted to the reduced form, NADH. No more glycolysis can run, and so it's oxidizing NADH back to NAD+. And in this case, there's usually a transition step, sometimes a transition molecule like acetaldehyde, but the end product in this particular one is going to be ethanol, 2-carbon molecule. And that's the one that was in the acid alcohol. That's why it was abbreviated ETOH. Alcohols always have a hydroxyl group on them, then that OH that was on there, and ET is the abbreviation for ethanol. So again, they do that because they need to run glycolysis some more. The alcohol, you already know, is a little bit toxic for them, but many times they live in environments where other organisms can take those alcohols and squeak a little bit more energy out of those alcohols and get just a bit more energy. Most of us don't consume that kind of fermented product, but a number of us eat beautiful Swiss cheese and other kinds of things, pickles. Some of us maybe consume that. Some of you use acetone to remove nail polish. There's lots of different fermentation pathways, and we can actually figure out which organism is which based on the products that they make in their fermentation. The products they make from the fermentation of pyruvate tell us a lot about the organism because it tells us what kind of enzymes they have. If they make certain products, they have to have certain enzymes. Some of you ladies, I got 40 seconds. Some of you ladies go and you've got these cute little bottles that live somewhere in your bathroom, usually maybe on a vanity somewhere. You take and unscrew the lid and you tilt them upside down and you get a little bit of that, or some of them have an atomizer or a mister that you spray somewhere on your body to smell really pretty. Most of those fragrances are actually because we've taken bacteria and train them with new enzymes to make different fragrances. So next time you mist yourself or dab it behind your ear, just remember that you're hoping that that guy that you're looking for really appreciates microbial waste products dabbed behind your ear. Okay? We'll come back on Thursday and talk about the rest of this.
Did that happen to record?